I want to start off by, welcoming everybody to, the inaugural scientific roundtable for the PKD-RRC for this academic year. Just as kind of a reminder, please visit our website if you get a chance. We have new exciting resources that are coming online, including a whole series of polycystin 1 antibodies that I think a lot of people will find useful. So a PKD2 missense patient variant is available up there now. So I encourage you to go up there, leave us comments on resources you think would benefit your research program. We'd love to hear about them, and then to consider how we can help support your research program. And with that, I'm going to introduce today's speaker, who actually needs no introduction, Dr. Greg Germino, one of the trailblazers in the PKD field for many years. And he is now a deputy director at NIDDK. And he will be telling us a story about PKHD1 mouse and why it does not have a renal phenotype. So, Greg, I'm going to turn it over to you and look forward to hear what you say.
Well, I'm not sure I'll be able to explain why it doesn't have a renal phenotype, but I'll tell you some unexpected things. So it's a little bit outside of probably where this community normally thinks, and certainly outside of what my laboratory usually thought of as area of interest. But I hopefully, in the process of characterizing this today, you'll see why we pursued it. And so let me just put on my slideshow here. Okay, if I can find it. Okay, here we go, and put it in presenter mode. Great, here we are. So
looks good.
And again, I want to just begin by thanking you for the opportunity to present some work that we've been working on this for some time. It's taken a little time to kind of work through some of the complexities, and I'm happy to share it with you today. So just as very briefly, and most of you, I'm sure, all know about autosomal recessive PKD. This has been an interest of the laboratory going back decades. You know, it's an uncommon disorder. It's 1 in 20,000 live births. It has a range of different presentations, both in terms of severity, in terms of organ systems. As you know, up to a third of individuals born with this condition may die within the first year of life. So there can be really some severe cases. And of course, there are those cases that really don't present until adulthood with respect to any kidney disease, if they have kidney disease at all. But the liver disease is usually a universal phenotype. And that's something we've also, as I'll talk about in a moment, has been seen in mice. On the right is showing some of the other sort of manifestations of the disease. Seems like it's on a time delay here. So some of the other manifestations of the disease outside of the kidney and the liver, as you know, you might imagine with that, you'll have systemic hypertension. The pulmonary hypoplasia presumably is due to the, again, very large kidneys in kids in utero that results in oligohydramnios and, again, a compression of the lungs. And then, of course, pancreatic cysts, which are seen in some, but not commonly. And so years ago, when we had first, isolated the gene as part of a consortium with groups in Germany and around the United States, we had identified isolated PKHD1 contemporaneous with Peter Harris's group. And we found the gene had, in the course of our cloning of this, had a fairly complex pattern of splicing. And some of this, of course, you can say, well, they're just RT-PCR artifacts or whatever else. But some of these were actually clones that were pulled out of, in the old days, when we had to actually use cDNA libraries and plate libraries. If any of you can actually believe that, and even actually have to manually sequence them. We had a lot of clones that had a really complex pattern of splicing, as you can see here. And, as you know, the gene is very, very large, 67 exons, and give rise to the largest open reading frame. But as I said, we identified many, many splice variants. And then when we cloned out the mouse ortholog a couple of years later, again, very, very big gene, very similar gene structure, and again, found multiple splice variants. The gene is, as I said, it gives rise to about a 13 to 14 kb mRNA, and that gives rise to a 4,074 amino acid protein, as shown here on the right. You can see by the structure, 3,800 amino acids of this approximately sit at least external to the cell membrane. There's a single transmembrane spanning element. and then about a 200 amino acid C terminus that has a number of different domains, as you can see here. There's a ciliary targeting sequence, there's a polycystin 2, been described a polycystin 2 binding site, nuclear localization sequence, and a mitochondria targeting sequence. Some years ago, Junya Kaimori in my lab had shown that fibrocystin undergoes a complex kind of notch-like pattern of post-translational modification with a cleavage site that's shown here, the S1 cleavage site that results in this ectodomain being tethered to the stalk, and then again things that would activate its shedding, and with that shedding and release of the ectodomain, there was translocation of the C terminus to the nucleus. More recently, Feng Chen and his group at Maryland have shown that this also can target to the traffic to the mitochondria. So this is the overall structure of the protein. And now we have, as many of you probably know, the functional significance of the PKHD1 splicing has been controversial. Evidence in support of this comes from a number of different places. Of course, the original cloning studies that I just described, and then some work done by Lisa Gay Woodford's group, where they looked at using primers from the 5' end and 3' end of the gene, did some RT-PCR, and characterized the pattern of transcripts that were produced. And as you can see here in the kidney and liver, and in both embryo and adult, there were some different patterns observed with multiple splice, different size transcripts that were detected. On the middle here, where my pointer is, this is actually just a blown up version of the kidney here showing the different size transcripts that were detected. They then also looked at adult organs and found, again, across adult organs, some different patterns, particularly in the testis and the lung, with other size products detected. They went through and cloned a bunch of these from the kidney and, again, put together a catalog of different splice products. And as you can see here, they found, again, a variety of different transcripts that at least they detected and cloned in their through their methodology. And they had gone on and looked at around the splicing junctions of many of these alternatively spliced products and identified intergenic motifs that they thought might regulate the transcriptional complexity of PKHD1. Again, all of you, I'm sure, know that there have been many different PKHD1 models made, all of us hoping to have one that will have a reliable, reproducible, and consistent kidney phenotype. We had made one in back in, I don't know, 2007, I think, approximately, when we reported this Delta 3-4. This one had actually, in its early rounds of generation, actually had a cystic kidney phenotype. And when we crossed it with PKD1 at the time, in fact, we brought out the phenotype and got, moderately severe, but we never saw the early, really severe neonatal form that's been observed in humans. What is universal in all of them, of course, is the liver disease. And so all of the models that were reported here have all had liver disease with variable kidney disease. And it's been argued that perhaps it's the splicing and the compensatory splicing that might accommodate or might account for the mild severity. And in fact, in our model, when we did some RT-PCR, there were in fact alternative transcripts that went around the deletion and the stop codon. And in fact, in a paper was published by Karsten Bergman as group a couple of years ago, they looked at their very large database of patients, and they found, and they were looking at genotype-phenotype correlation. And as many of you know, the literature suggests that bilineal truncating mutations are typically associated in this condition with more severe disease. So they looked through their database and found some individuals that had, again, bilineal truncating mutations, and in fact, they were relatively mildly affected. And so when they characterized them, they found that, in fact, there had been splicing around the mutation, and they think that that might be that complexity, that splicing complexity could have perhaps have accounted for the milder disease than would have been expected in these individuals. Of course, there is, strong counter evidence to suggest that splicing is not functionally important. This is a paper that came out of Chris Ward's group a number of years ago. On the left is showing cellular RNA probed with, as you can see, different probes that go along the length of the PKHD1 gene. And you can see there are some additional transcripts that they saw, particularly with more 5' to middle of the gene probes. Here's the poly A RNA lane on the right here. So they had a clever strategy to see if they could test this functionally, and that was by generating another mouse model. And this mouse model they generated to have two different things they could test. One was to, in their minds, completely knock out the gene by introducing in the knock-in fashion a transcriptional stop site, so that the gene would be null in the germline state. And then under Cree recombination, they would pop out this stop transcriptional stop site, and then have knocked into exon 3 a V5 tag they could then use to track the amino end of the protein. And so this is the stop go. And so looking at the stop, when they looked at in the mutant state, you can see here all the transcripts appeared to go away. So whatever they were seeing here on the left went away in the knockout. The V5 mouse, where they deleted that stop and allowed the transcription to proceed, the transcript came back, and they corrected for, there was a loading problem here, so this is not due to the effect of the knock-in. They then characterized the protein using the V5 antibodies. So the V5 tag was knocked in here to the very amino end of the protein. And as you can see here in the kidney lysate, this is actually a membrane preparation. So this is a lot of protein, membrane protein. But you can see here there's a doublet here, around 500 kilodaltons. And in fact, when you treat it with PNGase, it goes down to a single protein. So this is the only protein that they saw with their amino terminus. They looked in the urine. They didn't see a lot of smaller products shed in the urine as well. And so they concluded that this was pretty good evidence that the splicing products are not functionally relevant. And I just want to bring you back to this slide just for a second to point out here in the del2. This is another model of ARPKD in mice. And this one, I want to just to kind of note that the transcript is preserved, and there is in fact a higher transcript, suggesting that there could be some transcriptional splicing around or alternative splicing that did go on in these animals to perhaps account for why there might not be such severe kidney disease. And in fact, the same was true in our del3-4, where we saw alternative transcripts by RT-PCR. So we decided to develop a two-step strategy to definitively settle this question. And that was really basically the goal is to just remove the entire darn gene, all 500 kilobases of it, and then see what happens. And so we decided to take advantage of the conditional line we had previously made that locks PKHD1 conditional 3-4. And the construct is shown here. So this is where the LOXP sites were flanking exons 3 and 4. So the idea was to make another knock-in line, but this one where the LOXP sites are flanking exon 67. And in this case, then we would have, and also at the same time, we'd knock in a tag to allow us to track the C terminus of the protein, and then we could recombine the LOX2B sites and then get a complete null by bringing them across alleles. So that was the vision, that was the goal. And this is studies that we did in collaboration with Terry Watnick's group and Patricia Oteda at the University of Maryland. So here is the schematic for the FLOX67 allele. As I said, we put in LOXP sites in intron 66 and in the UTR, downstream of the UTR quite downstream of the UTR for the gene. And as you can see here, we knocked in a triple 3HA tag into the end of exon 67, so we could again track the protein in vivo. And so this is what it would look like in a schematic of the protein. You can see here again, the HA tag was put at the very end. And if you successfully deleted all of this, what would happen is you'd take out most of the domains that have been described as being important in the C terminus. So we thought, we'll get some interesting functional readout on the relevance and functional properties of these domains. So we did the LOXP deletion, First, we characterize by the way, just characterize the line. That's in Patricia's paper. The references here. Very nice way to track the protein in vivo. She did some immunostaining and characterized the protein's pattern. So it worked well from that point of view. Unfortunately, when you knocked it out, as Lisa Gay Woodford likes to say, the mice were joking and smoking. They were just completely happy and normal. Here's a picture of the kidney. No change in the kidney size, no change in kidney structure. And in fact, the liver also was fine. So they had no effect in either organ. So these animals were perfectly happy and healthy. So we then said, okay, well, that's interesting. Let's see if we can now generate that recombined allele. And so this is just shown here what we were trying to do. So we figured in meiosis through recombination, these two loci or these two LOXP sites would be about 500,000 base pairs apart. So we thought maybe by recombination we'd be lucky, be low risk on low effort on our part. And so the idea was shown here. So after crossing over, we'd have at least one allele, one haplotype that would have both LOXP sites for exons 3 and 4 and exon 67 on the same haplotype. Well, this didn't work, as you might imagine. Recombination is not so high in mice. It's lower than in humans, and so this didn't work. And so we said, okay, can we use another strategy to force meiotic recombination? And that was, we found a Cree that is very active in germ cells. And so this is what we use. And again, the goal here was to have a crossover between the LOXP sites flanking exon 67 and those flanking exon 3, 4. And if things worked, what we'd have is here just exon 2 and then nothing, just the UTR, the end of the UTR. And so this strategy was actually incredibly successful. Almost a quarter of the offspring were heterozygotes with the right genotype. And so we were really excited and delighted that this worked. It's also a cautionary note that if you have any Cree activity in germ cells, any of your Crees are active at all in germ cells, and you have LOXP sites, no matter where they are in the genome, they may be doing some things you didn't know or didn't think about. So just to keep that in mind as a sobering reminder. So we did all the sorts of testing, RT-PCR, genomic PCR, everything across the gap, just to make sure that things were as we expected, because we were doing some funky things with the genome. And we also did whole genome sequencing just to be sure and confirm that there was the deletion that we wanted and there weren't deletions that we didn't want or anything else that went sideways in the line. And so this is just showing the genomic sequencing data. The mice were born at expected Mendelian ratios. And there was no observed perinatal lethality. So there's no splicing compensation that's protecting these mice from early demise. They were, again, they were born healthy. We looked at their kidneys and we aged them, trying to hope to see that we'd see something in their kidneys. And their kidneys were generally pretty normal. If you look at enough of them and you look at them as they age, in fact, the female kidneys are a little bit larger, even statistically when tested, they're a little bit larger, the mutants compared to the controls, but there was no obvious cystic change. And this is something that actually had been previously reported for some of the other PKHD1 models, that the female kidneys may have slight dilation of some of their tubules. And so this was not completely unexpected, and this is not a new finding, but just again point out, again, sex is a biological variable, and we did check for that here. In this report, it was again co-authored by Dr. Ishimoto and Menezes in my lab. We did do some transcriptional analyses, single cell RNA-seq of these kidneys, just to see if we could identify any compensatory pathways. We did find a few genes that were differentially expressed in different cell types, but really didn't find a whole lot of pathways that were dysregulated. And the only signal we found where there might some suggestion of rewiring was with ubiquitination pathways. Oops, let's see here. Why is this not moving? Here we go. Going on to looking at other organs, the liver, again, shown here is 15-month-old mutants. And there was a variability in severity, as you see. On the right, fairly severe one. On the left, a little less so, maybe more fibrosis. But this is, again, something that's been previously reported, so there was no surprise here. They also had other phenotypes that have been previously described, including ascending cholangitis, choledochal cysts. And what was interesting and not previously reported, but what was very, very penetrant in this line was dilated common bile ducts. As you can see here on the left, you can barely see it here, and on the right, it was clearly dilated, and that was consistently so. We also did some resin molds of the biliary system in these mice, and shown on the left is the wild type. You can see a beautiful branching structure. On the right, it appeared that the overall branching structure was intact, but you can see the distal branches are in fact reduced, and so suggesting again a diminished density of the secondary branches. Other expected phenotypes, as you might imagine, as we see in humans, there was hypersplenism, presumably from the kind of portal hypertension from fibrosis, and these are aged animals. And then, of course, we saw occasionally, very, very occasionally, ascites. We also saw occasionally a pancreatic cyst. This is a dramatic example of it. So again, but these were not common findings. But then things went sideways and took an unusual turn. And the first unexpected phenotype was that we had reduced male fertility. When we used male homozygous del367 sires, they had consistently low litter size. So we said, okay, well, let's try to boost this. And we went back to some frozen sperm and some sperm that we'd isolated from these males and said, can we do this in vitro? And again, to our surprise, even by in vitro fertilization, the sperm of the males del367 was, as you can see here on the right, was quite low and this was three different sets of studies that were done. And this was done again in real time compared to wild type. We then looked back historically at some information that we had before using sperm from these animals and compared it to our work done again with other PKHD1 lines. And you can see here that the del367 line has consistently always had low fertility rates compared to even our other PKHD1 line. So this prompted us to think back to an old northern blot from 20 odd years ago or something like that, when we were first cloning out the mouse gene. And this was a commercial northern blot that we had probed with a piece of exon 5 and then something around exon 41. And what was striking at the time, and we noted it but didn't know what to make of it, was this peculiar little transcript down in the testis of about 1 kb. And this, it wasn't contamination from plasmids or anything else in our laboratory, because this was a commercially purchased, clone tech northern blot back in the day when you did that sort of thing. And so this got us thinking, could there be some other transcript or alternative transcript, PKHD1 based, that's within the PKHD1 locus? So we went into various databases to see, again, what's been profiled for sequencing, transcript sequencing in sperm. And what we found here is that in differentiated sperm, as you can see here in the middle pile, there's kind of a low level of exon detection across the gene. But you also see is that an undifferentiated sperm and round spermatids, there's this very high signal of about 3 exons in the middle of the gene, around exon 40, that actually correlate with a long non-coding RNA called 4930486103 RICN. This is embedded within the PKHD1 locus, going in the opposite direction. If you blow that up a little closer, just to kind of expand that part of the picture, again, you can see exon 1, exon 2, and exon 3. And so this actually, I suspect, is what we were detecting here with our probe back 20 odd years ago. This long non-coding RNA has an inverse pattern of expression You look across, again, published databases compared to PKHD1, where PKHD1 tends to be high, this thing tends to be low. And conversely, in testis adult, here this thing is extremely high and PKHD1 is quite low. So we think this long non-range coding RNA probably has an important role to play in sperm maturation. Now, we didn't test this formally. One would have to go back in and knock out specifically one of those, those exons, and this is beyond the scope of what our interest is. And we didn't do any functional testing of the sperm to see why did they move weird, or did they, why did they didn't fertilize effectively. Those are studies for somebody who's interested in that primary problem. And of course, the mice could be available to anybody who's interested in that. But at least it's suggested that there is something embedded. There is presumably in this locus something that we knocked out that's accounting for this phenotype. And then we got to unexpected phenotype number 2, which is this eye malformation. Shown on the right and in the middle here is kind of the normal eyes that you see in mice. And here in our Del 367, you can see a corneal opacity. And actually, as they age, the eyes becomes atrophic. Pulling out the eye globes and looking at them, you can see here that, again, the mutants, the homozygous mutants, have enlarged eye globes. And this is a 100% penetrant phenotype. This is a very, very penetrant phenotype. Looking at these, again, animals as they aged, we're looking at eye weight to body weight at 1 week old. There is no difference. This is an acquired phenotype, but by 4 weeks old, as you can see, they clearly have a significantly enlarged eye size. This is somewhat variable over time, as you can see, as the animals age, as there's some scarring or whatever else. But you can see that they still, in general, again, eye weight stays large. We did look again by sex, and as you can see here, there is no difference by sex. And finally, we measured intraocular pressure in these animals at around 3 weeks, 4 weeks of age. And as you can see, intraocular pressure was in fact elevated in the mutants and not in the others. Glaucoma, which of course is a result of increased intraocular pressure, or one of the causes of glaucoma is intraocular pressure. And so we looked at, and one of the consequences of that is loss of retinal ganglia cells as they age due to the pressure. And so we looked at that in our both 1 week old and 4 week old mice. And as you can see here, no difference at 1 week old, but by 4 weeks old, a pronounced decrease, as shown here also on the right by immunostaining for a marker of retinal ganglial cells in the, again, in the homozygous mutants. As a reminder for anatomy, for those who don't know the eye, and that certainly includes me, this is, the lens, the anterior chamber is up here. So this is the lenses here, the pupils here, the fluid is generated inside below the iris. It comes through the pupil and comes here into the anterior chamber. So this should be a space between the lens, the pupil, and the cornea. Okay, so that's how it should look. So this is what it looks like in our mice. So here is on the right a 1 month old wild type, and as you can see, there's the anterior chamber, there's the iris, and there's the pupil, and there's the lens, and a normal ciliary body. Looking at the double mutants here, the homozygous mutants, what you can see here is that the lens is attached to the cornea. The iris is attached to the cornea. And you can see there's again, a rudimentary ciliary body in these animals. We went back in time, and again, looking at even by E15.5, you can already see that they are malforming. You can see that, again, it's attached here, and it's not here, better shown in a close-up here. And going back at 13.5, that's the last point that we see actually normal histology in the developing eye. So you have this eye phenotype. As you know, that's not something that's been reported in any other PKHD1 mouse models. We reached out to a number of groups with large ARPKD human data sets, and nobody had, again, reported consistently an eye phenotype that would be anything like this. So we thought, well, maybe like with the testis, maybe there is some eye-specific transcript that we've missed and not been aware of. So we looked in, again, published databases, both of the eye and various, again, single cell databases, transcript databases for eye and human and mouse. And they're shown here. And what you can see is here is in the, this is the corneal endothelial cells. This is where PKHD1 was expressed in humans to some extent. And we know that the interior chamber, that these are derived from neural crest cells, and we know that TFAP2B is an important determinant of neural crest cell fate and development. And so we looked at TFAP2B, and of course, we saw co-expression of TFA2B in the same cell types in the human. As you know, Lisa Gay Woodford's group had previously reported that TFAP2B is upstream of PKHD1 and controls PKHD1 expression. So this coordinated expression wasn't perhaps surprising. Whoops, sorry. Oh, where go? My finger's too quick. Here we go. Going back now to the mouse, looking at the mouse, again, TFA2B is expressed again in stroma 2, and stroma 2 is, again, this is a cell type that's heavily enriched for, again, anterior chamber elements in corneal endothelial cells, and this is where there was the highest level of PKHD1 expression, although, as you can see, it is also quite low. TFA2B is interesting because TFA2B, as I said, controls neurocrest cell development, and others have previously knocked out TFA2P2B in neurocrest cells, and in fact, they got an identical phenotype to what we're describing with the corneal opacities and the anterior chamber dysfunction. So this seemed like a possible cause of perhaps PKHD1 being downstream of TFAP2B and that causing this phenotype. So we looked to see if there was, in fact, perhaps a PKHD1 transcript that was specific to the eye or enriched in the eye that might be under TFAP2B regulation. And so we looked in the databases by exon specific patterns. And as you can see here, in fact, what we found is that there is an enrichment in the eye for distant part of the PKHD1 gene, exons between particularly 54 to 56. This was, again, a striking enrichment of exons specific for that distal part of the eye. Now, this is, of course, a library that was made with a 3' tag, so we can't exclude that is the reason why there's enrichment for distal exons. But in either case, we did also look nearby to see if there were any enhancer sites for TFAP2B, and in fact, there were some. So this suggested perhaps the idea here is that TFA2B is upstream, PKHD1 transcript here in the eye is downstream, and that would then cause a disease when PKHD1 entirely is deleted. Just as a reminder, TFA2B turns out to be actually close to PKHD1 on chromosome 1 in the mouse. It's about 700, 800,000 base pairs away. And this brings up an interesting side story, which is there was a report, a handful of years ago, of somebody trying to do something completely different. They were trying to make a transgenic model of something, and they knocked this gene in. They had a complicated outcome, and they got mice that had a phenotype just like ours and like the TFA2B mouse. And when they characterized the genome effects of this thing, they were able to rule out it had anything to do with the transgene. It had everything to do with the chromosome rearrangement that they had here, as shown here. It was a kind of a complicated duplication deletion that disrupted the area between PKHD1, which is over here on the to the right, and TFAP2B. And when they looked at the tissue of these eyes, they found decreased TFAP2B and TFAP2D expression in the eyes. And so somehow this integration here and disruption here disrupted this distal gene expression. So we looked at AFA2B or AP2B expression in the eyes of our mice. And as shown here, on the left is a control, on the right is DEL367, and this is at 13.5 days. Close up is here on the right. What you can see is AP2B staining is markedly decreased in the eye of the DEL367 mice. And you can know that the staining worked because you can see that there was, in fact, persistent staining elsewhere for AP2B. So this is suggest again AP2B is decreased in these mice. Now you would not expect if AP2B is upstream of PKHD1, and if the mechanism of disease is that del367 is the cause of the phenotype, you would not expect loss of AP2B expression in these cell types. It should be persistent. They just don't form properly because they need the PKHD1 to do the next step of the process. And as you can see here, AFP2B is in fact greatly decreased. So given the fact that AP2B is persistently is missing and shouldn't be, and given the fact that there was a complex chromosome rearrangement between PKHD1 and TFAP2B that gave the same phenotype, we wondered if there might not be, in fact, some sort of other global chromatin structure issue here that's controlling or that's causing this phenotype. And so we looked in again in published databases, and we in fact could see that both mouse and human PKHD1 are part of the same topologically associated domain. So there's genomic interactions between the PKHD1 locus on the right and the TFAP2B locus on the left. So this prompted us to say, to think, well, maybe what if we tested? Could we test this interaction by crossing our PKHD1 del367 mice to a TFAP2B KO mouse line, and then see what happens to the compound compound heterozygotes? And this is what we found. In fact, the animals were born at normal Mendelian ratios, but in fact, they had exactly the same eye phenotype with the opacities, the enlarged orbits. The weight of the eye to body weight was also enlarged. Their intraocular pressures were elevated, and they, in fact, again, also got reduced retinal ganglial cells as the animal aged. And this phenotype was, again, 100% penetrant. Looking again at these animals, a little bit further detail. Here's the staining showing that, again, the retinal ganglial cell line number is decreased. Again, staining for AP2B, absent staining, as we had seen previously, and shown here the histology at 4 weeks. You can see, in fact, that the iris is attached to the cornea. Ciliary body, again, is poorly developed, and you can see that the lens and the cornea are attached. So again, histologically, the same phenotype we'd seen in the del367 and the TFAP2B conditional mice. So interestingly, despite this, the complementation here, they do not develop renal cysts or hepatic cysts. So human PKHD1 and TFA2B are part of the same topologically associated domain. So this made us think about what could there be any, again, clinical relevance to this finding. And in fact, we would suggest that there might be. Looking in some published databases, this is the one of the sites that's sponsored by NIDDK. Used to be called, it's the Accelerated Medicine Partnerships Program. Used to be just on diabetes, but now it's on metabolic diseases more broadly. And here, PKHD1 actually is listed as one of the top 20 genes with SNPs associated with primary open-angle glaucoma. And so, so there's, again, genetic evidence linking variants near the PKHD1 locus to this phenotype. Here is the listing of those SNPs and their location, the different study where this association was found, and then the nearest gene that was reported in the literature for all of these. And you can see almost all of them are close to or within PKHD1 as opposed to TFA2B. Shown here in the bottom of the panel, actually, you can see, again, the chromosome structure. Here's TFA2B on the left. Two of the SNPs are in TFAP2B that are associated with POAG, but all of the other ones are here in within the PKHD1 locus. Down on the right is just showing the mouse PKHD1 where the what we did with our deletion. And these other X's show the site of where the other different mouse models have targeted in the literature. And none of them, of course, have had disrupted this entire structure. And so that perhaps explains why none of the deletions that were made previously were big enough to disrupt the architecture in a way that could account for the eye phenotype. Finally, just to close out, the third odd aspect of this story was that we found incisor malocclusion at a very high rate. Now, those of you who manage animal colonies know that incisor malocclusion is not an uncommon phenomenon, particularly in the CPK line, but you're usually talking about, 1%, something 0.5%, something, it's reproducible, it's reliable, but it's not frequent. Here we're talking about almost 100%, at least 100% penetrant phenotype. 96% had this phenotype, and the trans heterozygotes had this same phenotype. Slightly lower penetrance, maybe, but again, extremely high penetrance, suggesting that, again, there's an important role for TFAP2B in incisor development. And in fact, there's a report, I think a family, I think it's in Thailand, that had dental abnormalities that were associated with a TFAP2B mutation. So just to close out, this is the model of what we think is going on in the DEL 367 mouse line, and how we think there's this genomic relationship between the PKHD1 locus and the TFAP2B locus. We think, again, there's these, we can't exclude that there are just enhancer elements here that are being deleted, but we think it might be more related to the overall genomic architecture that's getting disrupted by this very large deletion, or by this very large, again, rearrangement that happens between these two genes. Shown on the right here would explain how we see the phenotype that we see. Of course, on the top with two wild type alleles, the cis regulation is intact. You have a perfectly normal, healthy mice. And the DEL 367 homozygotes, you have no cis regulation on both alleles. And so in both haplotypes, so again, there's TFAP2B is absent, but only in a very targeted subset of cells. It really speaks to just incredible precision of regulation of the gene, and again, how the GWAS studies sometimes tell us these things. And then finally, in the trans heterozygotes, where we have, again, on one allele, we have the DEL 367, so there's no cis regulation. And on the other allele, there could have been cis regulation, maybe, but there's no TFA2B to respond because it's been deleted. And so again, no TFA2B expression, and we get the phenotype. So in closing, I just want to, again, acknowledge the incredible work done by Yu Ishimoto, Luis Menezes, and Feng Zhao on this work. Again, I want to recognize our larger group here at the NIDDK. We are a small group, but this is, all of our group that we have right now. And I want to, again, acknowledge my collaborators on this work over some period of time. Again, Terry Watnick and Patricia Ateta at University of Maryland, and then a number of colleagues around NIH from NEI. As you imagine, we're not eye specialists, so we needed to bring in some specialists, and we have some experts from the neural crust development team as well. And so with that, again, thank you for the opportunity to present, and be happy to take any questions.
Greg, thank you for a wonderful talk. I'm going to open up for questions. I'm going to ask those that ask questions to raise their hand and then unmute. I know Jeremy had a question in on the chat, as did Celia Prince. So I'm going to start with Jeremy. To foster conversation, please unmute yourself and ask your question.
Yeah, thanks, Greg. I mean, you got into, after I asked my question, I think a much more compelling hypothesis than the one that I was envisioning 3/4 of the way through your talk. I guess I was kind of thinking about similarities between the eye and the kidney, and maybe even the efferent ducts of the testis. And so I was wondering whether you had looked at the trabecular meshwork in the PKHD1 deletants.
So we had not done that sort of sophisticated analysis of the trabecular meshwork as one, ideally again would. This is this was kind of like a side project. I mean, we had this like unbelievable, you can't just ignore this phenotype, right? It was a really dramatic phenotype, and we knew that there was some data in the literature to suggest that there was this relationship between SNPs in the gene And it's kind of an amusing story. I was at a meeting where they were presenting this AMP, database. And so I was like, oh, okay, they're showcasing this thing. And I put in my favorite genes to see what would pop up with associations. And there was PKHD1 for POAG. And it's like, what the heck? How could that be? That doesn't make any sense. I don't know anything about it. And that was before we actually had the eye thing ever show up. And so I circled back to it. And the literature has gotten even stronger over time, showing an association. So it is. But someone really, could and probably should do that sort of in-depth, really analysis of the eye. I want to say one other thing, which was another confounder for us as we were trying to work through this, is if you look, actually, IFT88, people have done a conditional knockout of that in neural crest cells, and they get this anterior dysgenesis. So that's why we really weren't sure. Is this a ciliary phenotype of PKHD1? It's a ciliary gene, a ciliary protein. I think at the end of the day, I don't think it has much to do with PKHD1 other than, real estate and geography, and probably more relevant to the TFA2B.
Awesome. Thank you.
Michelle.
Thank you. Beautiful work, Greg. I just wanted to ask you to go back to the original question about multiple splice forms. Even with this comprehensive null model, you are finding these extraordinary phenotypes, but you're explaining those phenotypes because of things other than the PKHD1 mRNA, right? The testis seems to be the link. The eye seems to be the DNA interactions. So the question is, what are your thoughts about additional mRNA transcripts in regulating, going back to the original question of whether there are other splice versions, and what are your thoughts about that after you've done a deep dive with this new model?
Thanks. So I think what we can say is there's very little evidence that compensatory processes from splicing have any role to play with the mild or absent phenotype we see in mice. I think that's a pretty strong conclusion we can make, because these mice are pretty normal histologically from a kidney perspective. And so I think, and that actually makes it maybe an interesting model to be testing for modifiers, because you don't have to worry about any sort of compensatory splicing potentially getting around your thing and maybe modulating disease. So here's a true null, and there's no question it's a null. Maybe Peter, I think, or Chris Ward might argue that his was a proper null as well, because it has the phenotype. But this, there's no question that there can't be anything that's coming at the splicing around it. In terms of, again, proving that it's the long non-coding RNA, or in this case, what we'd love to be able to do, but it's just, I think, practically speaking, trying to get the neurocrest cells to do the TAD, analysis on a small number of cells, that would be really difficult from these mutants. So I think that's the challenge that we face, but I think that we don't have good evidence that splicing has a lot of functional significance in the context, at least, of the mouse models. Now, in humans, does this provide us, as Karsten said, maybe a mild, modulating effect? Maybe. Can't exclude that.
Thank you.
I apologize if I destroy your name here, but Jubuka, is that?
It's people call me Luba.
Luba? Okay, sorry, Luba.
So great presentation, and I have to say, chromatin structure is my favorite subject. So my question,
I'm in trouble.
My question is about HNF1 beta relationship with PKHD1, because HNF1 beta mice also have renal cysts, and HNF1 beta also regulates PKHD1 transcription. So my question is that a transcriptional regulation or something, again, having to do with the TAD or overall chromatin structure of the locus? What are your thoughts about that?
I don't know. I mean, I think, for instance, even in, with the TFAP2B, right? I mean, mice get a cystic disease when you knock it out, but if they're working through PKHD1, they must be working through some other pathway as well, because PKHD1 by itself in the same strain doesn't give you that phenotype. So we tried to look at the TFAP2. We made some TFAP2B nulls. I mean, we got the line from groups that have made the line. And, we got one animal that survived the neonatal period because they all die from a variety of different abnormalities. And that animal had cystic disease, but it's very mild. And but it's in the same strain, and so in the other, we have hundreds of PKHD1 mice and don't get it. So the effects of, and I suspect HNF1 beta is probably the same. It must be operating through a network of genes that it's dysregulating in mice that result in the phenotype in mice if they're getting cystic disease in mice, because PKHD1 itself does not cause that phenotype generally in mice. And I think what's interesting, and what we tried at various times, very unsuccessfully to determine is, we know there's got to be a modifier of some sort, right? We had a mouse line, this del3-4 mouse line that I described in the beginning. That mouse line in the beginning, it was in a mixed background. It was a very mixed background. That animal did have cystic disease, and when we bred it to clean it up, everything went to heck. I mean, the kidney phenotype went away. So there's something out there that is discoverable that can result in these cystic disease in mice, but it's not splicing as far as I can tell. And I can't, with again HNF1 beta, how it's doing it, I don't know, but it can't be doing it just through ARPKD. I mean through PKHD1, because those mice don't get reliable cystic disease.
Thank you.
Martin Prince, did you want to ask your question? It was in the chat. The question was, it just moved on me. Where'd it go? Wondering if the infertile mice had seminal megavesicles.
So I don't remember actually looking. I don't remember the testicular structure, but I don't remember the animal collaborator, the vet who did the sperm retrieval for us. I don't remember them ever commenting on any abnormal histology when they did the harvesting. And in fact, I'm not sure that would account for the in vitro defects, unless it led to a functional consequence later, extra ex vivo. So I can't exclude that, but nobody ever, I don't remember honestly, because that was some years ago we did that. I don't remember specifically noting the testicular architecture. But again, the ex vivo studies, the in vitro fertilization studies, were the ones that demonstrated, again, consistently low fertilization rates.
Okay. All right. Any further questions?
Yes. I probably missed it. I saw in one of the slides that you showed that the mice had a incisor malocclusion.
Yes.
And is this the PKHD1 dependent phenotype, and why it is?
Yeah, so it was 100%, not 100, 90-odd% of the del367 homozygotes had the phenotype. And it also was highly penetrant when we crossed it with the TFAP2 beta mice. So the trans heterozygotes, the ones that had, again, they were heterozygous for the del367 and heterozygous for the TFAP2B mutation, they also develop the phenotype. So we conclude that it's likely through, again, the same mechanism of disrupting neurocrest cell function in the development of the incisors. And in fact, there's some literature to suggest that incisors again, neurocrest cells participate in their formation. So that's how we see it happening. We don't think it's a PKHD1 gene or a protein functional effect.
Sorry, go ahead.
So just one follow-up. So do you see this in other models? For example, in other PKD models, that you have been working with?
No, we don't see it in any other models. And I think because we've deleted the entire locus, and so we think this is causing disruption of the chromosome structure in a way that's disrupting a distal enhancer that affects TFAB2B expression in a subset of neurocrest cells. I didn't say this, but one of our collaborators, I didn't have time to show the data, but she actually looked with our team at TFAB2B expression at early stages at E8, E9, in that range. And all of the neurocrest cells do their normal things and do their right things, and in terms of their differentiation. And so it really is a specific phenotype later that is under AFP2B control. That's disrupted again only in a subset of isolated subset of neurocrest cells that give rise to in the periocular mesenchyme that give rise to the eye.
Thanks, Greg. A great talk.
Thank you. Thank you.
All right, we have time for 2 more questions. Jeremy and then Michelle.
So sorry to be greedy here, Greg. I was wondering if I could expand the conversation a little bit and get your thoughts about how you might arrive at a more faithful model of human ARPKD. For example, do you know of any co-expression in the mouse of fibrocystin with fibrocystin L? Could there be kind of overlapping functions with PKHD1 like one?
We looked at that a long, long time ago to see maybe, and they seemed like to be non-overlapping patterns, but I have to say I haven't gone back in the modern era when there's a lot more information and a lot better information to potentially look at that and exclude that. I mean, so I can't really answer the question, but when we looked at it before, it seemed that it would be an unlikely sort of process. But, you can't exclude that when in the absence, what we haven't done is looked at our PKHD1 mutants and seen if there was upregulation or altered regulation of PKHD1L in the non-cystic kidney that could be compensating. And that would be something we could easily do. We probably should do that just to exclude that. But my guess is that's probably not the explanation.
Got it. Thanks.
All right, Michelle, last question.
Okay, so me too, like Jeremy being a little greedy, Greg, since we have you here. Now that you've conclusively proven that the locus is null, there is no compensatory splice and all of those things, to explain for the lack of the kidney phenotype, what are your thoughts of why is it that the mouse does not get kidney cysts? I'm sure you've thought of other things now that now that the transcript issue is finished.
Yeah, no, that's why we did the single cell studies. We were hoping we'd find some pathway that was upregulated that was compensating for the loss of PKHD1. And it was really underwhelming in terms of how informative it was. And so we don't really have a good explanation. It's like many things, one of the things I find really remarkable, we think about things being conserved, right? Genes are conserved. We think networks are conserved, pathways are conserved. What I find, the more that I look into these things, it seems to me, although the parts may be the same, how they work together is often different from species to species, even when we think in conserved pathways. And so I don't know how those parts are working in the context of the kidney in the mouse and why it's redundant. There's a redundancy, obviously, and there's something that can be done to bring it out, whether it's knocking out PKD1, which maybe makes the phenotype a little worse, or there's some other compensatory pathway that obviously we haven't identified, and love people's ideas about how to find that.
All right, Greg, I want to say thank you for a wonderful talk, stimulating ideas. I think the video for this will be up on the website probably in the next week or so. So if you want to go and watch it again or share it with somebody, please feel free to do so. Thank you for stimulating questions. And Greg, once again, thank you for taking the time to talk us through the phenotypes in the PKHD1. Wish you would have told me why they don't get cysts, but hey, that's for another day, right?
Next year.
Thank
you very much for the opportunity. Thanks for the great questions. I really appreciate it.
Thank you, guys.
Have a good one, everybody.